Welcome back to Hidden Wisdom Academy Biology. In today's lecture, we'll study the anatomy of the stem in flowering plants. We'll learn the primary structure of dicot and monocot stems, how secondary growth happens in dicots, and how these features help in transport and support. This topic is very important, especially the differences between dicot and monocot stems. So, get your notes ready, and let's get started. As always, in this slide you can see the syllabus and PYQ topics with their years, so you know what to expect in your exams. Let's talk about primary structure of dicot stem. In a dicot stem, the outermost layer is the epidermis, a single protective layer with a cuticle and sometimes hairs or trichomes. Below it is the cortex, which has collenchyma for support and parenchyma for food storage. The innermost layer of the cortex is the endodermis, also called the starch sheath because it contains starch grains. Beneath it lies the pericycle, made of sclerenchyma that provides strength. The vascular bundles are arranged in a ring, and each is conjoint, collateral, and open, because cambium is present between xylem and phloem. At the center is the pith, which stores food. A common example of this structure is the sunflower stem. Now, let's talk about primary structure of monocot stem. In the monocot stem, the outermost layer is the epidermis, covered by a thick cuticle for protection. There is no clear cortex, endodermis, or pericycle. Instead, there is a uniform ground tissue made of parenchyma cells. The vascular bundles are many and scattered throughout the ground tissue. This arrangement is called a tactostele. Each vascular bundle is conjoint, collateral, and closed, meaning there is no cambium and no secondary growth. A sclerenchymatous bundle sheath surrounds each bundle for support. The pith is not distinct because the ground tissue is continuous. Maize stem is a typical example of a monocot stem. Let's compare the anatomy of dicot and monocot stems. In dicot stems, vascular bundles are arranged in a ring and secondary growth occurs due to the presence of cambium. In contrast, monocot stems have scattered vascular bundles, no cambium, and hence, no secondary growth. These structural differences explain why monocot plants like maize remain herbaceous, while dicots like sunflower can form woody stems. Here is a labeled diagram showing the differences between dicot and monocot stems. In the dicot stem, notice the ring of vascular bundles with cambium present between xylem and phloem, which allows secondary growth. The pith is clearly visible in the center. In monocot stems, the vascular bundles are scattered throughout the ground tissue, with each bundle surrounded by a sclerenchymatous sheath. Cambium is absent, so secondary growth does not occur. The pith is not distinct, and the ground tissue is undifferentiated. Understanding these diagrams is crucial for NEET practicals and theory questions. Let's talk about functions of stem. The stem has many important functions in plants. It supports leaves, flowers, and fruits for photosynthesis and pollination. It transports water and minerals from roots to leaves through xylem, and moves food from leaves to other parts through phloem. It can store food and water, like in potato tubers. The stem grows too. Apical meristem gives primary growth, and cambium in dicot stems allows secondary growth or thickening. Stems also help in vegetative reproduction through runners, stolons, and rhizomes. In some plants, like cactus, stems can even perform photosynthesis. All these functions are vital for the plant's survival, growth, and reproduction. Now, let's talk about practical observation in stem. In practicals, a dicot stem like sunflower shows an epidermis with cuticle and collenchyma underneath. The cortex is visible, and the endodermis or starch sheath surrounds the vascular bundles, which are arranged in a ring. Each bundle is conjoint, collateral, and open, with cambium between xylem and phloem. The pith is at the center. In a monocot stem like maize, the epidermis has a thick cuticle. The ground tissue is uniform, and vascular bundles are scattered, each with a sclerenchyma sheath. Cambium is absent, and the pith is not distinct. Let's practice few questions to test your knowledge. Let's start with question 1. Which of the following statements about dicot and monocot stems is correct? Option A. A dicot stem has scattered vascular bundles, a monocot stem has vascular bundles in a ring. Option B. A dicot stem has cambium between xylem and phloem, a monocot stem lacks cambium. Option C. Monocot stem exhibits secondary growth, dicot stem does not. Option D. Both dicot and monocot stems have pith and distinct. Explanation. Vascular bundles are in a ring in dicots and scattered in monocots, so that statement is wrong. Dicot stems have cambium for secondary growth, making them open, this is correct. Monocot stems lack cambium, so their bundles are closed. Only dicot stems show secondary growth, so that statement is wrong. The pith is distinct in dicots, so that statement is also wrong. So, option B is the correct answer, that is, a dicot stem has cambium between xylem and phloem, a monocot stem lacks cambium. Now, let's move to question 2. In the dicot stem, the vascular bundles are Option A. Conjoint, collateral, and open. Option B. Conjoint, collateral, and closed. Option C. Radial and open. Option D. Scattered and closed. Explanation. In dicot stems, xylem and phloem lie on the same radius, which is called collateral. Xylem is inside near the pith, phloem is outside near the epidermis. The bundles are open because cambium is present between them, allowing secondary growth. In monocot stems, bundles are closed and have no cambium. Dicot bundles are in a ring, not scattered, so B and D are wrong. They are collateral, not radial, so C is also wrong. So, option A is the correct answer, that is conjoint, collateral, and open. Before we wrap up, let's quickly look at the common mistakes you must avoid in stem anatomy. First, never confuse the vascular bundle arrangement. Dicots have them in a ring, while monocots have scattered bundles. Second, remember that dicot stems have cambium and can show secondary growth, but monocots cannot. Also, don't mix up the pith, it's distinct in dicots and absent in monocots. Another key point, monocot stems have a bundle sheath, which dicots don't. Avoid these mistakes, and you'll score full marks in this topic. That is it for this lecture on the anatomy of stem. Remember the key points. Dicot stems have vascular bundles in a ring with cambium, allowing secondary growth, whereas monocot stems have scattered bundles without cambium. In the next lecture, we will explore the anatomy of leaves. We will study transverse sections of leaves, epidermis, mesophyll, veins, stomata, and some special adaptations. Also, don't miss our daily NEET MCQs and PYQ series and biology podcast for quick revision. See you in the next lecture.